Xtalks connects professionals in the life science, medical device, and food industries with useful content like webinars, job openings, articles, and virtual meetings to help you succeed in your career. This podcast brings together some of our editorial staff to share insights into the latest B2B industry news to help keep you up to date.
Hello and welcome to the Xtalks Life Science Podcast. I'm Ayesha Rashid, Senior Life Science Journalist at Xtalks.com, and this week I'm delighted to introduce a very special guest on the show. We have with us today Dr. Howard Berman, who is the Chief Executive Officer and Chairman of the Board at Coya Therapeutics, which is a clinical stage biotech company developing regulatory T-cell therapies targeting neurodegenerative, autoimmune, and metabolic diseases. In 2021, Dr. Berman founded Coya Therapeutics, and Dr. Berman has over 18 years of entrepreneurial and industry experience working at the interplay of science and business. His experience offers him a unique combination of business acumen and scientific credibility with the ability to assess, quantify, and bridge both disciplines. Dr. Berman gained corporate experience with increasing responsibilities and positions as a Medical Science Liaison at AbbVie, where he spent 7.5 years launching Venetoclax in CLL and later supporting numerous solid tumor assets, including serving in leadership roles at Novartis, Pharmaceuticals, and Eli Lilly as well. To learn more about Coya Therapeutics and its regulatory T-cell therapeutic programs, let's turn to Dr. Berman. Dr. Berman, thank you so much for being on the show today.
Thank you so much for having me.
So I'd like to start off by asking you more about Coya Therapeutics and what led you to found the company, and if you can provide an overview of the company's main missions and goals.
Thank you. Well, Coya was born out of a tragedy, actually. My father, brilliant man, triple board certified physician, one of the smartest people you'd ever meet in your life, started to have what we now know as a cognitive loss, and that decline started to escalate and speed up, which is what you typically see. So I pulled some strings and I got him to see the best of the best at Houston Methodist in Houston, and Dr. Stanley Appel. And at that meeting, Dr. Appel said, "Look, Howard, unfortunately, there's not much one can do for dementia now. However, meet me next week because I'm working on some technologies and therapeutics that are very exciting and may actually play a role in ameliorating and alleviating dementia and other diseases." And the following week, I was there meeting Dr. Appel, and he showed me some technology and some data and some science that was unbelievably exciting. And that's how Coya was born. We spun off his therapeutic and technology into a new company called Coya. And 3 years later, we were going public. And it was the week that we went public that my father passed away. Unfortunately, he wasn't able to get onto our treatments, but
sorry to hear
set
that.
the groundwork. Yeah, thank you. So that's how the company was born, and it was born with Dr. Appel. We did this hand in hand, and now we've become a public company, and we're on for the journey to change the way patients are treated.
And so as you ventured into developing therapies for neurodegenerative diseases like dementia and Alzheimer's, how does Coya's approach intend to overcome these obstacles? And so far, what have been some of the biggest challenges in developing therapies in this area or in this space?
Yeah. Well, I think one of the challenges and difficulties has been understanding the field's understanding of the mechanism of action of what is happening in terms of the pathophysiology of these diseases. It's critically important, thanks to the research that we've done, that inflammation has really become a key driver. Inflammation in multiple aspects, both in the early genesis of the disease, but also the subsequent decline of these patients. And companies now are realizing that inflammation plays an important role, but they've been targeting only one or individual pathways in that process. We've taken the next step and we're targeting multiple pathways simultaneously, but we're doing that also through this pathway called the Treg, the regulatory T cell. That's a critically important cell type that no one has been targeting in these neurodegenerative diseases, and now we are leveraging for our approach.
And so could you tell us a bit more about this Treg approach in targeting the pathophysiology of neurodegenerative diseases? And I know that you have a lead product candidate, and we'll talk a bit more about that, but how are you utilizing Tregs to target, again, the pathophysiology of the disease?
Yeah, so Tregs are first, as a background for the audience, they are a type of cell in the body that is responsible for controlling inflammation. It's the most important cell that if your immune system ramps up, the Treg sort of pushes the immune system back away. And what we've discovered is that the Tregs are dysfunctional and they're compromised in multiple neurodegenerative diseases. They're not working properly. And because of that, the level of inflammation in the blood, if you measure it in these patients, ALS, Alzheimer's, Parkinson's, it is exceptionally high. And in ALS, it's off the charts. So what we're doing is we have biologic drugs, meaning proteins that you can simply inject like a insulin injection into the skin. And what they do is they act to enhance the Tregs, convert the Treg dysfunctionality to functional status, and also at the same time slow other inflammatory processes that are involved in causing Tregs to go back dysfunctional. So by doing these things together simultaneously, you have a synergistic effect on the Tregs, but also on other very bad inflammatory processes.
And so could you tell us a bit about the specifics of the technology platform that led to the development of one of your lead product candidates, which is Coya 302, and how that developmental pathway has been executed, and what pipelines you have that are looking at 302, of course, in neurodegenerative diseases. Where is the development at right now in terms of clinical trials and all of that?
Sure. So I want to start in the beginning, where the first series of experiments that we did was we took patients' blood and we measured their Treg function in ALS patients, and we then followed them out for many years, and we found that the patients who had poor Treg function statistically significantly were dead much earlier than those patients with good Treg function. That was the first series of experiments. The second series is we took patients' blood therapeutically, we isolated their Tregs, we enhanced their Treg function, and we gave it back to the patients in 2 clinical trials and in ALS patients. And we showed that the patients, we could stop progression in those studies or significantly lower the progression of the decline in these patients. And that was a very remarkable and interesting finding. But we realized that unfortunately, cells are expensive to manufacture. They also can convert in the blood. If you put live cells back into people, they can convert from functional to dysfunctional. So we asked the question, was there a way that we could not use cells, but use proteins and other biologics that are off the shelf that could enhance and ramp up the Tregs inexpensively and even in ways in which the cells can't do? And the answer was yes. We found the pathways and the drugs that could do that. So we started using multiple drug combinations that we were testing preclinically, And we found that the two that were the best, when you put them together, dramatically enhanced the Treg function. And those included low-dose interleukin-2, which is a protein, but it's at a very low dose of this cytokine. And we have the intellectual property and the formulation for low-dose IL-2, and that enhances Tregs. And then we combined it with abatacept, or CTLA-4 IgG, which is a fusion protein that blocks other inflammatory pathways. And we put the two together, and that combination is dynamite in terms of its effect on Tregs and inflammation. And the final step was we put those drugs into patients with ALS. We also tested in Alzheimer's disease, and that's where we saw the dramatic effect in ALS patients. And in the cohort of patients we tested who were declining prior to treatment, the patients stopped progression. At 6 months and at 12 months, they had hardly any decline whatsoever. That is a remarkable finding. It's an early finding, but it's leading us to now conduct and file an IND for a large randomized clinical trial, which we hope to provide meaningful data to read out by 2025.
That's excellent. That's wonderful. And so those studies were done in ALS. Am I right? And so are you looking at these drug combinations with the Treg targeting biologic in other neurodegenerative diseases as well? So dementia, Alzheimer's, Parkinson's?
Yes. So we call this Coya 302.
Coya 302, yeah.
Coya 302 is low-dose IL-2 and CTLA-4.
Okay,
IgG.
So that's, yeah, I was actually a bit just thinking about, like, is that the actual biologic? But I'm like, I don't want to ask a dumb question.
Right.
There you go. Okay, so it's a fusion of the IL-2 and the CTLA-4.
So it's the low-dose IL-2, and it's separately the CTLA-4, and they're given as in a kit.
So
they're provided separately. They're not put into one formulation. They're separate drugs, and they're done. That's done because of the different half-lives of the drugs.
Yeah, makes sense. Okay.
But the 302 is we call it a pipeline in a product.
Yeah.
Because we're testing it in ALS, and we're testing it in frontotemporal dementia and Parkinson's disease and Alzheimer's disease. And that's really important because the similar mechanisms that drive these diseases are found in these different conditions involving dysfunctional Tregs, heightened pro-inflammatory cell types. And we've also discovered biomarkers in the blood that you can actually monitor potentially the progression of the disease, but also how the drugs may impact those biomarkers to affect the disease.
That's great. And so you mentioned that there are blood-based biomarkers. Are there also any imaging biomarkers? We've seen a lot of focus on different imaging biomarkers in the space. So are there any of those kinds of biomarkers or any other types that can serve as readouts or surrogate markers for monitoring the efficacy of the drug?
Yes. In ALS, imaging is not specifically used, at least brain imaging is not specifically used as a marker. But in other diseases, like in frontotemporal dementia, yes, you can actually look at using PET scans at the brain and look at inflammation and reduction in inflammation, same as in Alzheimer's disease. There's ways to look at certain types of something called TSPO, PET scan binding, and look at the inflammatory reductions in the brain. And in fact, we've done that in a study in Alzheimer's disease, showing pre and post treatment, and that's proof of concept showed a reduction in inflammation in the brain in a patient that we measured. And in an upcoming trial funded by the Gates Foundation, for one of the components of our 302, which is the low-dose IL-2 component, and that's reading out very soon in the next 2 or 3 months, a double-blind randomized trial, we are doing imaging in every patient, both the placebo and the active, and that should be able to determine how the drug works to reduce the inflammation. That could be a surrogate marker, in addition to other things that we're measuring in that study.
And beyond neurodegenerative diseases, of course, inflammation is a hallmark in many autoimmune diseases as well, and metabolic disorders. And I think your company profile mentions that you are also probably moving into these spaces as well.
Yeah, autoimmune disease is a critically important area with a high unmet need as well. Autoimmune conditions are driven by dysfunctional Tregs as well, and inflammation. It's the body's way of attacking itself, unfortunately. You see it in lupus and in scleroderma, and it's a very exciting field, and it's an area that, as again, it has tremendous need for new therapeutics, right? So the ability to target both Tregs, but also other inflammatory pathways, both the B cells, the humoral response, but also the adaptive immune response and the innate immune response simultaneously may play a beneficial role. The abatacept component of 302 is approved already in rheumatoid arthritis and in graft versus host disease. So imagine us being able to put low-dose IL-2 with the CTLA-4, the abatacept, that potentially could play a even more beneficial, a further beneficial role, a synergistic role in these other autoimmune diseases as well. So we're excited about the role in the future for those.
And so what kinds of changes have you seen happening, or what are, I hate to use the word trend, it just sounds a bit kind of pop culture-y, but in the space of neurodegenerative diseases especially, we know that there's still a great need to better understand the pathophysiology of the disease, but at the same time, we've been hearing a lot in the last couple of years and been seeing approvals for various drugs, monoclonal antibodies, some controversial, more controversial than others. And so how have you seen this space evolve, and how do you see it moving forward?
Yeah, that's a loaded question, but it's a great question. And it's specific to each disease. Every disease is different. I will say for ALS, Lou Gehrig's disease, it is one of the highest unmet needs out there. And one of the problems and limitations of other drugs, companies, if you will, has been that their drug, no one knows, they don't know how their drug is working. And there is no defined mechanism of action. And that's a problem, because if you're not able to determine how your drug is working, how can you shout out to everyone that if there is a beneficial effect, that you're having an impact on the target? Our drug works specifically on known, defined, delineated mechanisms of action, and we target multiple pathways. And that's unique and differentiated from what these other companies are working on. The other area is biomarkers are becoming a critically important area in ALS, but in other diseases as well. And as you know, in oncology, biomarkers have driven the field, and the FDA is very interested in biomarkers, and we are leading the way in understanding new and novel biomarker and biomarker trends longitudinally, how the biomarkers change over time. And that, I think, is also going to allow us to make strides and innovations with our therapy that others haven't been able to make. And we recently came out with a data showing a very strong correlation on a biomarker for survival in ALS. But we're doing the same sorts of things in Parkinson's and in Alzheimer's, and that data in the future should come out. So sort of taking a playbook from the oncology field is, yes, inflammation is important, but how can you target multiple pathways, and how can you use and leverage biomarkers?
And so you mentioned that the FDA is very interested in biomarkers. What is the regulatory landscape like now, especially for biologics? I know the FDA has a separate arm for evaluating new drug applications for biologics. And so, and especially since COVID, things have changed quite a lot on that front. So how is the landscape now and navigating it with a new product candidate?
Yeah. So the FDA is very supportive, particularly in the field of rare diseases, and they have a mandate and a mission to move treatments forward that help those patients. And in terms of biologics, yes, they are, it's a separate division, but they are also vested in the success of companies, and they work very closely with companies. They are focused on the same things that you would anticipate. First of all, safety and tolerability of the therapies, but also the manufacturing of the products. They want to make sure that what they call the CMC, which is the manufacturing, is up to par and up to standards. And if the drugs do actually work, that CMC will be approvable according to FDA standards. So you have to have that aligned. But I have to tell you, it's been a pleasure to work with FDA. They're very collaborative. They work hand in hand with you, and they provide excellent advice and suggestions. And we've only had very positive interactions with them.
It definitely is a partnership. I think in the past it wasn't really regarded as such, but I think the FDA also has sort of been more welcoming and to really serve as a partner to companies and developers and people innovating especially for rare diseases and all types of diseases, of course, oncology and other areas. So that's really great to hear firsthand. And also in terms of speaking about partnerships and collaborations, we've also been seeing a lot more collaborations between academia, industry, government organizations, advocacy groups. And so how important are those in today's drug development landscape? And particularly when we talk about ALS, what comes to mind are things like the Ice Bucket Challenge that was very popular and a big thing almost, I would say, a decade ago. And I've spoken to people in the field, and they've firsthand been able to tell me the impact of that in terms of funding and getting their drugs through even to market. So it's been really wonderful to see. So how important are those partnerships, collaborations, and patient advocacy included in all of that?
Well, it's very important. The Ice Bucket Challenge was a viral outcome for patients. And I think the fact is that really launched this Lou Gehrig's disease, high unmet need out into the community.
Yeah. Public awareness, right? Yeah.
The awareness, the awareness, and also the need for funding and support and advocacy. The patient advocacy groups are very vocal, and they need to be that way because the patients deserve and their families deserve new treatments. There's just been, unfortunately, a host of symptomatic treatments, no disease-modifying treatments. And that's across the board, not just for ALS, but for other neurodegenerative conditions. These are terrible diseases with just, unfortunately, a litany of failures that across the board. So it's important and it's critical. So we started the relationship and collaboration with academia, and we continue that. We innovate with our relationship with Houston Methodist and with other academic institutions, and that's the way that you can save money and save time and do some innovative work and do it very quickly and efficiently, particularly when you have a company, a smaller company, and you're wanting to make progress without the need for huge expenditures, and do bench work and also translational work. That's what we innovate, and that's why we're successful, and that's why we have accomplished so much in such little time. The parallel, again, we work with advocacy communities, but we are very careful. We don't want to overpromise to these patients. We want to underpromise and overdeliver. That's our mandate, and that's what we do, not just for them, but for investors and alike. And by working with all of these groups of people and collaborating, as we talked about before, that's the way that we can have a synergistic relationship with everyone and achieve the success that we were out there to achieve, and run clinical trials in a manner that will get the patients enrolled and get the data out there, and hopefully have a treatment available for these unfortunate patients and their families.
Indeed. And I really like how you said you hope to over deliver, which is great. And the other thing that I'm gleaning from this is that you've been on both sides. You've been on the patient side with your father, and you're on the development side, trying to develop new treatments. So you really, I think, are very well positioned in terms of doing what you're doing. So that's really, really wonderful to see, professionals like yourselves in the field. And hopefully we'll be hearing about the next breakthrough treatment from Coya.
Yeah, thank you very much. I'm coming at this from both the individual perspective as well as from the business perspective. So I'm looking at this in a way that many other founders and CEOs don't necessarily have. It's a meaningful and it's a personal journey for me.
Yeah, no, absolutely. And that's so wonderful to see. And I have one final question here. And this is just talking about the biotech sector as a whole. And in terms of it rapidly evolving with advancements in genomics and AI, of course, and personalized medicine. So how do you foresee integrating these new technologies, or is it too premature to have AI and robotics in your labs and in your studies now? But what's your take on adopting and adapting to new technologies?
One always has to adopt new technologies and be open to ways to leverage these breakthroughs. I find that AI is actually highly intriguing and actually becoming a central player in drug discovery and also innovation.
Seems like it.
Now, there are ways to incorporate AI, and we're looking at ways we can use AI not just in how we run clinical trials, but also in other permutations of combinations of drugs. When you combine for example, our low-dose IL-2 component, are there other drugs that we can use in combination?
So like an in silico approach kind of a thing.
Right. So imagine, you could take AI, you could put it into modeling out the best inflammatory pathways that you can target simultaneously and come up with the best of the best, which it would take you potentially a long time to just go into bench research, which are the best combinations. But if you could somehow use algorithms or AI, if you will, to do that could save a lot of time, energy, and generate new intellectual property. So that's ways in which you can do that. But there's other ways as well. And we intend to leverage that, but also genomics and also single cell analyses, which is the ability to look at how individual cells are performing and how the treatment may be modified by single cell analysis. So there's all new technologies that are creeping in, and we we are using our relationships with academia to leverage those.
And yeah, you mentioned academia again, and it just takes me back to grad school. And I really wish we, as students and even students now, it would be great for them to have exposure to industry and to really, sort of see those collaborations, which when I was in grad school, I didn't see any of those collaborations at all. But it's great that those academic collaborations are important to you and that you are engaged in them. And I think that that's very important, and maybe we're going to see more and more of that going into the future. So, just lastly, I'd like to know more about sort of your short and long-term goals at Coya. What can we expect in the coming months or the next 2 to 5 years?
Yeah, well, in the short term, we're filing an IND, and we'll have that opened hopefully soon, within the next few months, for our phase 2b ALS trial for Coya 302. We hope that trial is going to be meaningful for patients. It'll be a randomized double-blind study, and we are excited about that study. We are reading out our data this summer for the randomized double-blind Gates Foundation funded study in Alzheimer's disease. That could be a strong catalyst and driver of the company. That will be, of course, we don't know the data yet, hasn't been unblinded, but we're still we're planning for that as well. And then, of course, for the next, we're working on in Parkinson's disease as well. We have a study coming out, an animal study coming out. We're planning an IND for an FTD study, which is happening this year as well. So we're going to do a clinical trial there. And a lot of that data for FTD and ALS will come out next year in 2025. So there's a slew of data. We have a lot of innovation internally. We're running at full speed in order to get things done. And we don't, here at Coya, we don't waste time. I can tell you that. We find this to be a mission that is, again, meaningful, but we need to get this out into patients and hopefully show the differential benefits that we're expecting or that we want.
Indeed, meaningful and must be timely because urgent unmet needs definitely in this space.
Correct.
Dr. Berman, I'd like to thank you so much for your time once again, for being on the show.
Thank you so much.
All right, that's the end of this episode of the Xtalks Life Science Podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, for joining us, and we'll see you next week. Bye for now.
If you enjoyed our discussions today, please share the episode with your friends and colleagues, and be sure to subscribe in order to be notified when a new episode is released. To join in on the discussion, you can find Xtalks on social media, email podcast@xtalks.com, or comment on the articles directly. Links are in the show description. Take a moment to join our community at xtalks.com to get access to everything we have to offer, including webinars, job listings, virtual meetings, articles, and more. The views and opinions expressed in the podcast are those of the speakers sharing them. They should not be taken as professional advice and do not necessarily reflect the policy or position Honeycomb Worldwide. For further information, email us at podcast@xtalks.com. Thanks for joining us, and we'll see you next week.